same answers. And the idea there is if you get your blood drawn in California and the sample gets split and goes to Northern and Southern California, 2 different labs, are they both going to say that you have cancer or not, right? You don't want to have one lab say one and one lab say the other. So this working group that I was a part of was called the Verification Working Group. And verification is like a softer way of validating something. So we wanted to put it through the due diligence of what a method validation would be like, but we didn't call it validation because technically it's not. Validation is very rigorous. Anyone who works in the pharmaceutical industry knows validation is a mind-numbing thing. It will take a long time to truly validate a method for robustness, ruggedness, reproducibility, sensitivity, and so on. We didn't go that far, which is why we're calling it verification. But we had a lot of researchers from different centers across the US, and this was the second study that we had done. The first one was published in 2009. I think I have some references to it in here. And this one was published in 2015. There we go, sorry, okay. Thank you, sorry. So this slide goes over the premise of why we're doing what we're doing, and I had brought this up yesterday. We wanted to go and shoot for that middle section where we are verifying the meaningfulness of potential biomarker candidates, screening a larger number of samples to see if there are any statistically significant abundance or quantity differences between different sample sets, and then take those, the ones that are statistically significant, and move them on to clinical validation. And so here we're using triple quadrupole technology in SRM and nanoflow. We were looking at plasma, peptides in plasma. So the technology, again, this slide has been used a lot, but it goes through the process and the workflow that we started with, which was first identify your candidate biomarkers, second get a signature peptide, which is unique only to that protein sequence, have one synthesized because you want to be able to do quantification. You want the quantification to be good, spike that into your sample, and then use, so I should also mention, a lot of this happened before the Q-Exactive and the HRAM boom happened. And so, so there was no PRM. There was no alternative to triple quadrupole technology. This was pretty much it. It was known to be the fastest and the most sensitive. And so that's why this platform has existed for so long and will continue to exist. So anyway, we all have gone through this. You spike your peptides in, you digest your sample, you run it on a triple quad, and you look at the peak area ratios. You know how much of your heavy you spiked in, and now you can calculate how much of your light is present. So these were the key features we were looking to evaluate in this large study. This study had 13 different labs involved. Ultimately, there were 15 total instruments. Some labs had more than one. And we were looking for testing these features of reproducibility. We needed to make sure retention times were steady, that all labs could get steady retention times using nanoflow, which that's not an easy task. Transferability, can we make sure that one lab gets the same answers as the other lab with the same exact sample set? Can we do this quickly? It's difficult with nanoflow. Can we get to sensitive levels? Because most of the interesting biological proteins that are doing anything are down at low nanogram per mil levels. Can we be accurate? How do we be accurate? How can we minimize subjectivity? Because up until this time, Skyline hadn't really been used mainstream. It was something we needed desperately, but many people were relying on vendor software packages from whatever instrument they used. And so the other question is a Thermo software calculating the data the same way as a SciEx software package? And then a statistical pipeline. Again, there wasn't really anything straightforward and easily accessible to the non-statistics person. I only know enough statistics to get me in trouble, and then I go talk to a statistician. They're like, oh, let me do this for you in R. I'm like, okay. So trying to get all this together was very useful. Yeah, so hold on a second. Oh no, it's a PDF. Shit. Okay. That's all right. Okay. How do I get it to present again? Sorry, Lindsay.
Sorry.
How do I get it to present from PDF?
View full screen.
Full screen view, thank you. Okay. Okay. Sorry about that. This is covering up a part of the workflow that was just more repetitive, but the point here in this study was we wanted to extend what we had done previously. So we worked with isotopically labeled peptide standards that were synthesized, the tryptic version, but we also had access to these fully N15 labeled proteins. So every nitrogen in the protein had an N15 label, which means the mass for every peptide, the heavy version, was variable depending on how many nitrogen it had. So heavy peptides containing arginine can be heavy by 6 or heavy by 10, and lysine can be heavy by 6 or heavy by 10. Those are the main forms for those. For these, our peptides didn't have a fixed 6 or 10 mass shift. They were dependent upon the amino acid sequence and its length, essentially. So that introduced some minor challenges, but shown here to illustrate, the N15 labeled protein has those little red dots, meaning it's fully labeled at every nitrogen in there. And then when we digest it, the idea here is we have a protein standard now, and it should behave very similarly to the light version of the protein present in the sample. We also had access to these unlabeled proteins. So we could set up a very well-controlled experiment where we would spike light peptide in and heavy peptide in. The heavy would be the internal standard. And then down lower behind this, you can't see it, we also added in the synthetic peptide standards. So here we had 35 proteins we were monitoring, a large number, as many peptides, up to 5 or 6, that I believe 5 that we could per protein. I guess here we had 10 participating sites, 15 instruments, and those covered thermo, water, SciEx, and Agilent. So our previous study, we monitored 10 peptides. Here we monitored 135 times 3 for the most part because of the 3 different forms. And the aspects we wanted to look at when generating this method was we wanted to characterize the protein standard. We wanted to look at transition selection and have it be something that was specific to each platform. We're not going to take a transition list from a thermo instrument and assume it's going to work exactly the same on a Q-trap that's from SciEx, because they do things a little bit differently. So we wanted to make sure it was optimized on a platform by platform basis. We looked for interferences, auto interferences. We tried to minimize retention time shift. We evaluated digestion conditions using different denaturants and designed a curve to calculate LOD and LOQ. And then also tried to help with automated data processing. So these are the protein targets. The ones on top they were selected ahead of time, and they were all thought to be cancer relevant. And then the control proteins shown down at the bottom, those are the ones from our previous study. We didn't have N15 forms, heavy forms for those. We did have the heavy synthetic peptides, tryptic peptides, to monitor. And so these, instead of having the light and heavy protein forms, we just spiked these as a fixed spike in all of our samples, and these were our quality control proteins. So even though they're light, they're actually not really relevant to much. They were just easy proteins to get. So it's something you can do if you spike it into all your samples. It can be a reduction, alkylation, digestion control. So this is kind of going through what we were talking about yesterday. First, we had our standard proteins. We digested them, and we saw what peptides looked good. And so we were able to show also any differences between the N15 and the not heavy labeled form. Moving on, we also wanted to qualify our standard and make sure our standard was reasonable. Now, N15 isotopic incorporation in a protein is always tricky because every N15 site has like a 99% purity or labeling efficiency. That's at the atom level. But if you take all of the nitrogen atoms that are present in your peptide, then it's not going to be 99% anymore. You're going to have the chance to have under-labeled forms of your peptide. And so that's what we did here. We digested the proteins and we looked at actual MS data in the blue trace. And then the observed data was it's digitized form of the observed, and we had a software developer that was able to adjust the percentage of N15 incorporation and adjust that isotopic pattern. So for those of, to orient you, for those of you that may not understand, this was done on a time of flight instrument, so we're getting intact peptide. This is a doubly charged, and so this is the monoisotopic mass of the monoisotopic ion of the heavy labeled precursor, and this one that's before it, usually don't see this. This is the underlabeled. So this is contribution from N14. And this is pretty significant. It's more significant than we see for synthetic peptides that are labeled with C13 and N15. And so what Mike was able to do was sort of adjust and simulate the percentage of isotopic incorporation, compare it to the observed, and found out that most of the peptides were pretty well labeled. We had some that were not so good, but overall these turned out to be decent, you know, knowing that they are not the exact number we spike in, knowing that there's going to be a small bit of imprecision and inaccuracy in what we're doing. So this was the layout of the experiment where we had the light and heavy proteins. The heavies were spiked in a fixed amount. The lights were spiked in a response curve. We selected these and put everything through Skyline. We had each instrument platform pick the top 5 product ions that worked best for them, then narrowed it down to 3, and then did collision energy calculations. We didn't optimize these. We didn't find it necessary because everyone's collision energy regression equations were pretty good. And then the final transition list, we had, if we just monitored the light and tryptic heavy, the synthetic heavy, we had 738 transitions. And then when we looked at the protein form as well, the heavy protein form, we had over 1,000. And this was done in 2010, 11, 12 or so, and this was challenging. The instruments were not as fast as they are now, and so this took a lot of care and headaches. So one of the first things we did was gradient development, and we looked at 4 different gradients. This one on top, the green one, was from our previous study, and that's what the lab, when I joined the lab, everybody used that gradient for their triple quad method. And I was like, that is just, So I took the heavy peptides and tried to optimize the gradient. And so the top one is the original gradient, and all of the peptides came out here, right? So like in a 10-minute window, we're running for 90 minutes. That is such a waste of time. And so the other gradients were just adjusted to see if we could spread them out, because remember, we're not just seeing these peptides come out here. Everything's coming out there. All of your plasma, anything else in your sample, it's all coming out in that 10-minute window. And I've had discussions with people saying, oh, but the peaks are taller. Like, yeah, but you could be getting ion suppression. The reproducibility isn't as good. The peaks aren't wide enough to get enough points across them. And we saw that from the previous study. But to give you an idea, so if you consider in terms of duty cycle, then this is sometimes why people run tandem columns where they can have the mass spectrometer acquiring useful data for as much of the run as possible. Here, the first run had only an 11-minute window where peptides were eluting over that 90 minutes. It's a 12% duty cycle. It's a huge waste of your mass spectrometer's time to be just collecting useless, nothing data during that time. And so as you separate everything out, we were able to get down to about a 38% duty cycle, which means you're actively getting good data 38% of that entire time, which still is not ideal, but that was the best we were able to do with nano. Because you can see most of the time is taken up by that wash and the reequilibration, and the mass spectrometer has nothing to do at that time, so we just kept it going. So the complexity of our targets, this was a study that was done on the cusp of when people were beginning to do larger numbers of peptide quantitation, and there were firmware limits in some of the instruments where they couldn't do more than 1,000 transitions in a run. So we had to go to SciEx and get a patch. We had to show them, like, here's 1,000, but we had to cut some of our targets because your software won't let us do it. And so they fixed it, and now most instrument vendors can have up to 30,000 transitions in a single method. So it's very important. If you don't get what you need from your instrument, talk to the vendor and they'll make it work. So this was a plot Brendan showed earlier during the IRT. How do you schedule? I mean, obviously, a peak is something that you want, and you want it to be good quality, so you'd like to keep the retention time window as narrow as possible, so you're not wasting time collecting data when a peak is not eluting. However, you can see on the top plot, this was over just 3 injections, we had a peak that was just shifting ever so slightly forward and forward. And you can't integrate a peak that you can't see. You can't integrate a peak that's beginning to get cut off. I mean, you can, but it's just gonna impact your data quality. So we had to make sure we were scheduling as narrow as possible, but as wide enough to accommodate retention time shifting. And the problem here is that, I mean, how many of you are able to log into your instrument overnight and watch it run to make sure you're not getting peaks cut off? Like I am. I slept with a laptop next to my bed and I kept waking it up with my foot. I'm like, oh, I got peaks, all right. And if you don't, then you have to stop it and update the retention times, or in the morning, you have to prepare to stop it, update your retention times, and rerun everything the night before that didn't get good data. And so we brought this up then to say something has to be done. And now there are things like isotopic standard triggering, that if the instrument sees something at a certain threshold, it'll have a contingency and trigger on other things. So you can imagine you could put in your heavy peptide to trigger on your light peptide, because your heavy should be there at a good abundance all the time. And right now, Thermo has a feature where it will change the retention time on the fly, much like IRT, where you have landmark peptides. During acquisition, if it sees your landmark peptide shifting, it'll shift everything in between landmark peptides. And so we're getting there. It's becoming easier, which is great. So again, why schedule? Obviously we had to, because there's no way we could get 1,000 transitions per second on the instruments at that time. But this is from a data set I recently acquired where we had the same cycle time. So we're going through a list of transitions in 1.8 seconds. My chromatographic peaks are about 30 seconds wide, so they're a little fat, but it was good because I could have a longer cycle time. If we ran it unscheduled with the 467 transitions or whatever it was I had, it was between 400 and 500, I was spending only 2 milliseconds per transition. And then when I scheduled with a 2-minute window, now the number of targets at any given moment is much, much less. It's changing though, because we're gonna have, like here, we're gonna have way more targets if you schedule in the middle of the gradient than at the beginning or at the end. So that's where you'll have the lowest dwell time. But here, what I'm showing is for a concentration curve and looking at the peak area ratio, percent CV, so this is light versus heavy. The blue is from unscheduled runs. So this is the precision that we're measuring these peptides at. Same sample, same LC, same gradient, same column, same day practically. And all of the scheduled ones that have higher dwell times are much, much more precise. So the variability decreases as you're able to spend more time acquiring the ion flux. Okay, so how many people have the best digestion recipe for digesting proteins and refuse to look at anything else? Because it's awesome. Oh, come on. You guys are all lying. Everybody has a favorite digestion protocol. I mean, everyone has a protocol in their lab. There might be 2 different ones in a lab, but people are like, it's like getting raised on this is what you do, and you don't really question it because everyone else in your graduate lab or your company or your lab has done it for decades and it works and oh, it's fine. Well, we had to question this, partly because urea works great as a denaturant, but it's a salt, it's icky, it can carbamylate your sample. We were worried about, is there any way we can make this easier for people? So DOC up here is deoxycholate. It's a salt that precipitates in the presence of acid. So we would denature with this deoxycholate mixture, and then after reduction alkylation, I think before adding the protease, we would acidify it and then spin it, take the supernatant off, and the salt crystals we'd throw out, and that supernatant had our reduced and alkylated proteins that we added the trypsin to. And then after that, there really wasn't any salt left in the sample because we were able to we didn't add any other than that deoxycholate. And then trifluoroethanol is another denaturant that it's an organic solvent, and we were able to dilute that out, and that again did not require desalting, which was nice. But when we looked at the percent recovery of our peptides, so these are all different peptides from different proteins on the x-axis and the percent recovery on the y, we saw that there really wasn't major differences between any of the protocols, with a couple minor exceptions. So you may see, how does this work? Like this purple one, these two purple ones, they happened to be much better recovered with trifluoroethanol, TFE, but everywhere else, that TFE protocol didn't really do so well. So are we going to change our protocol just for those two peptides? Those are the only two peptides you care about, then maybe it's worth it. But in general, the other methods seem to be fine, so we just kind of stuck with what had worked. This was a poster at ASMS. I have a PDF if people want it, I can make it available, but you can also go to the ASMS website and it's up there. Okay, so LOD and LOQ calculations. One question is, over what concentration range should you set up your curve? So one is, if you have the biological information and you know what level of the analyte needs to be present within the range, then make sure you span that range and a little bit on either side. Most of the time when we were designing these experiments, we wanted to make sure that we would go low enough that we would spike levels well below the LOD, so we could demonstrate it was not detected there or here, but as you go up higher, now you can see the signal show up. So what's shown here is a response curve. I plotted it on the log scale. It's often very convenient to do your dilutions on a log-based scale, because when you plot it on a log scale, you can see them equally spaced out. If you do it on a linear scale and plot them linearly, you'll notice that all the points get crunched up at the low concentrations, and so it's just difficult to see. I personally do all of my regression analysis and slope determination on a linear scale. I do not like the log scale, because remember, we should always on a linear scale get a y equals x slope. So here we were throwing out numbers, and this was something. Oh, so this is the method that we use to calculate LOD. This is something that Dr. Armani will discuss later. It takes into account the signal, the average signal present in a blank sample that has everything except your analyte. It has your heavy, it has your background, it has any other things that you would spike into your sample except the analyte itself. There's a factor that allows for a confidence interval sort of measurement and other statistical stuff that I'm not good and qualified to tell you well. It's got the standard deviation of the blank, and then a standard deviation of, so this is standard deviation of the signal of the blank, in the blank, and then of the next lowest spike level. So, and then that's the square root of the number of replicates you made. So here, this actual equation doesn't require a calibration curve. So that's kind of nice, but it's always good to determine your linear range, so we went ahead and we're doing that anyway. And we knew we wanted to get down to the attomole on column level, and we knew we should get up to the hundreds of femtomole on column level. We didn't go higher because we didn't want to have to deal with carryover. And if you have a biomarker that high, like, you don't have to, that's just awesome. And we knew we didn't need to go lower because at the time you really couldn't see lower than attomole on column using nanoflow in our techniques. So initially we picked a 16-point curve for just a couple of sites to evaluate to make sure that people using 4,000 Q traps and people using 5,500 Q traps, which are two different generations of instruments from the same company, one is more sensitive. We wanted to make sure that the 4,000 wasn't going to only detect the top 3 points, and the 50, we wanted to make sure it was fair for everybody. So the reason we picked 16 points and we did that was because the previous study that I had just gotten involved in had again a 9-point curve in the blank, and the blue dots are the proposed spike levels for that study, and the scale here is picomole per microliter. So that means we only went down to 1 femtomole, which wasn't that impressive. So you can see in our response curves, everything's really linear. Nothing falls off that line down at the low end, even on the linear scale. So we wanted to make sure we didn't make that mistake again, and we wanted to make sure we went down low enough. So this was the 16-point curve, and we made sure we had adequate representation of all the different vendor instruments that were doing these analysis. And the LOD calculation that was on a couple of slides back was used to determine the median LOD is a black bar, and then the interquartile range is the bigger box, and outliers are shown in black dots. And so this is just an example here where, so most instruments were good. They had a really low and similar LOD level with a pretty tight interquartile range, except this SciEx 5500. So it was a later generation. It should have been more sensitive. It should have been more precise, especially compared to a 4000, but it wasn't. So this was something that we wanted to investigate. This is also the first study where we incorporated a system suitability protocol, and we'll get into that tomorrow in detail. But the point here was every 8 hours we would run a system suitability run. It had nothing to do with our actual sample. The gradient was even a little bit different, but it monitored peptides using nanoflow, and we wanted to make sure that we got just the consistent performance out of those peptides from run to run, and we bracketed them every 8 hours to make sure that we wouldn't go a whole day and find a failing system suitability and then lose a day's worth of data. So here, everyone had to run the system suitability first before beginning the assay and do a bunch of replicates to establish reproducibility. So that 5500 Q-trap that had the high range of LODs had pretty good CVs, and the 4000 Q-trap also did too. But when you look at the median LODs from those instruments, you're like, wait, but the 5500 should be much more sensitive. It should be down to the low animals, not 220 animals. So then when we went and gathered all of their system suitability data and plotted percent CV of the peak area versus, these are all the different peptides that were monitored over the entire course of the entire setup of running the calibration curves, we found that the CVs were 70%, and the 4000 had much better CVs of 30%. What we know is that variability plays tightly into the measured sensitivity of a method. And so if your instrument isn't giving you very precise results, then the sensitivity will suffer simply by calculations. So this system, also I should point out, the way these curves were run were singlicate low to high. We had a QC sample in the middle, system suitability every 8 hours, and then each curve was followed up by a set of blinded samples that the instrument operator didn't know what the concentration was, and they were told to run them in a specific order, and they were quasi-randomized so that there would be a high concentration followed by a low, by a medium, and scrambled, so that we would make sure that if there were carryover effects, we could address them and understand them. And so what had happened here was over the course of those 4 curves, the variability of the instrument just began to tank because the sensitivity was dying. And ultimately, they got their instrument replaced because it just didn't survive after this for some reason. Also during the study, we found that Skyline was there and it was working great, but there still were, the features that you see today were not all implemented. And so we were working through the process of, Brendan, I need to see this, I need this present. And so one of the things that we asked for was being able to notate when not to use a peak because it was cut off, because there was variable electrospray stability, because there was a bad injection, any of a number of reasons. And so there was a do not use checkbox, and then something that you could say about why not to use that, because you would need to disqualify everything. And so when people were able to go through and notate their data, this is the same set of data where this is initially just being pulled into Skyline and processed, and then somebody actually going through and making sure the data, the peaks were picked correctly, the data looked good, any bad runs were disqualified for a reason and notated. It dropped, again, their LOD by half. And so, having the ability to do this is very important, because you can't just trust without looking that all of your data files are fine, right? We ran, and we had 125 peptides times 3 different versions, and so of heavy, light, and 15 labeled. So this was a lot of data to go through, but at this point, it's hard for me to say, trust the software. I like trust, but what's that? Trust, but verify. Thank you. Glad you guys know. So one of the other goals of this particular study was to compare it to our previous study from 2009 and show that things have gotten a lot more sensitive. So there were some modifications we made to the protocols. The first was in 2009, which is the Adana study. It's in Nature Biotechnology. We used neat plasma, which is just plasma spun out, and that's what you get. For this study, we used depleted plasma. So the top 14 most abundant proteins were depleted from the sample, which means we concentrated all of the other peptides. Now, we never actually tried to detect anything endogenous here. All of our light peptides were spiked over a concentration range. And so the question is, like, how did we make it more sensitive? And so Steve Carr and I always have this argument, and I don't really argue with him because I don't want to argue with him, but in my head, I'm like, you're so wrong. So, sure, we did sample enrichment, and we concentrated the sample, but we still spiked peptides in to less, to plasma that's still complex. And I think the effects that are more important here are that we optimized the gradient. We spread all those peptides out, so there was less potential suppression in the ionization, and we were able to get better measurement across our chromatographic peaks. And all of our operators that were working in this study, this was pretty much their second iteration. And we had gone through training, most of them during the first study, about how to set up nanoflow connections. This is before NanoViper was around, before IonKey was around, before it got easy. People were using the silica blades to cut fuse silica and make finger-tight connections for high-pressure LC systems. And so it was a big challenge back then, but we had very extensive protocols, and that's some of what I sent out in the email yesterday. So you can see our digestion protocol and our run order and our concentrations and everything. But I think the biggest factor, aside from doing that depletion, was like culminating everything that we had learned from previous studies and optimizing the gradient. Okay, so at the time that this was published and we were able to present on it, this was the first large-scale inter-lab study to look at quantitation where we had protein standards. We know that the proteins helped improve the accuracy of our measurement because we knew how much of a protein we spiked in, and having it there for digestion took a lot of the variability from digestion away from the whole experiment. We did have a sensitivity improvement. We had to do retention time scheduling, which we didn't for the previous study. And then here we just pointed out that software and computational methods needed to catch up because we went from tens of peptides to over 100 peptides, and it was an exponential jump, which was really challenging at the time. And then having this data quality filtering, so having the ability for someone to go in, and this is where the summary plots came out in Skyline, where you could see if a peak wasn't integrated in the right place or the wrong peak was picked, you could look at the retention time plot and like, oh, that's not the right peak, and fix that one instead of having to manually go through everything. So instead of actually having to go through every single file, we were able to look at the summary plots and pick the files that looked wonky and then make note of them. And so again, at the time, the request to vendors was to have data-dependent MRM-MS, so something that if a peak is there, trigger contingent peaks. And we're now, years later, getting there, which is wonderful, but it's just like, why does it take so long? Retention time updating or prediction of systematic drift, this is something that Skyline now has integrated into it. We needed column heaters. We had to have all of our labs purchase them because we saw one lab, this is Fred Hutch, they had an HVAC system that shut off overnight. And so when you plotted their retention time over the course of a 24-hour period, they had a sine wave because between 6 a.m. and 6 p.m. and midnight, like there was this perfect chromatographic shift that went back and forth. It was because they had construction going on in their building, and they went and spoke to their facilities and like, you have to do something to this room. And then we got them column heaters, and the retention times were fine. So all these little things that you have to pay attention to, lessons learned. So in the grand scheme of things, I just wanted to wrap this up with like, yeah, that was a lot of details and things that you may never do. And what's the whole point of this? You know, at the end of the day, we're trying to look for ways to diagnose diseases, especially things that don't show up until very late stage, because there might be things happening in your body that you can't get from plasma or you can't access easily. And so my vision is to go away from biopsies and invasive sample acquisition and tissue punches and things, and evolve what we find and validate or verify by mass spectrometry for point of care tests. And this will help doctors make earlier decisions on treatments. This will help people make moment-to-moment decisions on, any kind of condition they may have during the course of a day. And more data will give you better control and an improved quality of life. And so this, I thought, was an interesting thing to bring up, because back in the 70s, if you had type 1 diabetes or elevated glucose levels, you had to go to a doctor to find that out. There was no way to test your blood. You would probably get thrown into jail for having blood in public. I don't know. It's just not what people would do. And now these tests are available for $1 at home. You can buy them without prescription. You can just prick your finger and within 5 seconds what your blood glucose level is. And that's not how it was originally developed. There was a lot of research that went into something that's now like a little strip that costs $1. And then from that, which was more public in the 80s, it's developed into real-time monitoring. So now every 5 minutes, if you wear a little sensor that's attached to a transmitter, it sends a signal to an insulin pump or some kind of receiver or your iPhone that tells you every 5 minutes what your blood glucose level. That's a biomarker. That's awesome. So if you think about it, what we're doing here, what we're trying to set up and validate is something really important, and it has this huge potential impact. So anyway, I just wanted to point that out, because I think sometimes we get so into my chromatographic peaks are shifting and skyline, but there's a real reason why we're doing this. So anyway, this was a very large undertaking. These are all of the people that were involved in some way or another with the CPTAC studies. And Birgit Schilling at UCSF and Buck Institute and Steve Hall at UCSF were co-chairs of the working group with myself over the last 2 years of the study. And so if there are any questions on that, we've got a break now, and then later we'll be doing a little more hands-on work with Skyline, as well as the LOD calculation lectures. All right. Great. Any questions?